DNA replication, with a focus on the complicated lagging strand.
Hello! Today we're going to be talking about a summary and overview of DNA replication with a focus on the lagging strand, since, as you can tell, it is a lot more complex. This is a close-up view of the left side of the replication bubble, and the direction of replication is going from right to left. Also, from the 5' end to the 3' end on the leading strand. So, as you can tell, I have the single-stranded binding protein labeled, the parental DNA, which is right here. That's the helicase, again, single-strand binding protein. DNA Pol 3, primase, primer. DNA Pol 3 again on the lagging strand. DNA Pol 1, notice how it comes after Pol 3, even though it's 1. Keep that in mind. It might try to trick you. And then this is DNA ligase. Notice how the leading strand and lagging strand both are being formed in the 5 to 3' direction. 5 to 3, 5 to 3, 5 to 3. Alright, so breaking down the lagging strand, here's a closer look at the lagging strand, and the first step that the lagging strand goes through is a primer. As you can tell, the primer joins the RNA nucleotides into a primer, right here. Notice how it says RNA. DNA consists of C, G, and AT, when RNA consists of C, G, and AU. So instead of T, they have U. So this is the template strand, 3' end. This is the 5' end. So the order of replication is being made from the 5' to the 3'. The primer is doing its job. And then this is a primer for the leading strand, and that is the origin of replication. Alright, so after our primer joined the RNA nucleotides into a primer, right there, it forms this little fragment for fragment 1. So the DNA Pol 3 polymerase adds DNA nucleotides to the primer, forming Okazaki fragment 1, which is in blue and labeled 1. Again, 5' to 3'. I couldn't stress that enough. You have to remember that. So that is what's binding, or adding the DNA nucleotides. So instead of the U, it is adding the T, along with, well, it's binding it to the A, and then it's doing the G and C again. And then, oops, my phone. Sorry about that. And this is the slider, just kind of comes before the DNA Pol 3. And again, that's the origin, and that is the leading strand. So the second step of the lagging strand. Alright, so after the DNA polymerase 3 does its job, which is adding DNA nucleotides to the primer, after that's formed, it detaches itself after reaching the next, this guy to that guy or whatever. I shouldn't say guy, but RNA primer to the Okazaki fragment. It detaches itself and gets ready to start the process over again to form a fragment 2. Alright, so fragment 2, this one, as you can see, has another original. So, that is the RNA primer for fragment 2, and fragment 2 is primed, then DNA Pol 3 adds DNA nucleotides and detaches again when it reaches the fragment 1 primer. So after this fragment 2 reaches this, the DNA Pol 3 detaches itself again. So that is the process of making the second fragment. Over here on the first fragment, the DNA Pol 1 replaces RNA with DNA and adding nucleotides to the 3' end of fragment 1, as you can see there. So the fifth step of the lagging strand is fragment 1 and 2. About fragment 1 and 2. Okay, so the DNA Pol 1 replaces RNA with DNA adding the nucleotides to the 3' end fragment 2. So this is kind of just like a more visual than that, so you can see what's actually going on here. And see, there's a little space right there. So it actually combines and adds the nucleotides. Again, remember, 5' to 3'. They are going anti-parallel. Now to the last step. The last step in the lagging strand is DNA ligase. DNA ligase forms a bond between the newest DNA and the DNA fragment 1. So as we saw before in the little slide or picture before, these were pink, and now they're blue because they changed to DNA. They changed to DNA nucleotides. And then the lagging strand in this region is now complete. That's the origin of replication. And the overall direction of replication, we know, is going right to left. Because you have to keep in mind that if we had a leading strand above here, where my finger is, it'd be forming 5' to 3'. So 5' to 3' again. Alright, so I know I just threw a lot of topics at you guys and a lot of interesting words, so here is a little table explaining each of those drawings I had before. Some of them might change the color because it just looks nicer. So, helicase is this little guy right here, and its job or function is to unwind parental double helix at the replication fork. So this happens right, if we're looking at here, right here. All right, and then the single-stranded binding proteins, their function is it binds to and stabilizes single-stranded DNA until it is used as a template. So those are very important. Topoisomerase. It relieves the overwinding strain ahead of replication forks by breaking and swiveling and rejoining DNA strands. So its job is to basically make sure that the parental strands aren't getting too swiveled up together and help them from actually breaking and not being able to form either a leading or lagging strand on there. So they're like kind of the controllers of they stabilize the parental DNA. Primase. This synthesizes an RNA primer at 5' end of leading strand and at 5' end of Okazaki fragment of the lagging strand. So, just primes it up. Basically just like puts little RNAs down. DNA Pol 3 in front of Pol 1. Don't get confused. This function is using parental DNA as a template. It synthesizes new DNA strand by adding nucleotides to the RNA primer or a pre-existing DNA strand. So, as I stated before in my drawings, sorry, I'm a little shaky, it puts DNA instead of RNA. So it changes those pink to blue. Speaking of color, DNA Pol 1, it removes RNA nucleotides of primer from 5' end and replaces them with DNA nucleotides added to the 3' end of adjacent fragment. So there we got the 5', 3', 3', 5'. The little DNA Pol 1, and there's the space there, because it's got to remove those suckers and put them into DNA and combine. Then DNA ligase. It joins the Okazaki fragments of lagging strand on leading strand. Joins the 3' end of DNA that replaces primer to the rest of the leading strand DNA. So there's a nice little drawing. I hope that this has been helpful for you guys. Here's a little overview of what we just went over. And I hope by watching this video, you have become more informed and understand the complex lagging strand of DNA replication. thanks for watching. Bye.
Just wait, there's more.
Oh, and before I forget, we talked a lot about the RNA and the DNA, and if you guys don't understand what that is, here we have the T binding with the A, which is thymine binding with adenine. And then we have the guanine bonding with the cytosine. And a difference between those two is that they have 2 bonds up here. The T and the A only have 2 bonds, and then the G and the C have 3 bonds. So that is something to take note of. And then this is the, as you can tell, the phosphate group, and that's the sugars. And you want to make sure that you know that this is a group, and this is a group. Do not get confused. This is not a group. This one is. So that is the DNA nucleotides. So instead of having T and U, they have T. No, shoot, I'm sorry. My apologies. Instead of having A and T, RNA have A and U, but they still have G and C. And then here's a closer look at what each of those nucleotides looks like.
Thank you so much for watching. I wish you the best in life and the happiest of days. Good luck on your test.